Hi everyone, so we are now on the second part of our discussion about the structural and functional organization of cells. So again, these are the different learning targets for this lesson. So again, important reminder, form fits function. Remember, every part of the cell, every organelle has its corresponding function. Okay, so now we will be discussing the different cytoplasmic inclusions. So first off, we have the ribosomes. Now, organelles called ribosomes make the proteins needed by the cells. Now, group of ribosomes function like an assembly line, helping to make proteins from amino acids. And ribosomes are found suspended in the cytoplasm or bound to membranes. Next cytoplasmic inclusion, we have the centriole. Now, the centriole is important because it participates in the formation of the mitotic spindle, and the wall is composed, or the centriole wall is composed of 9 groups of parallel subunits arranged in a longitudinal fashion. And each of the 9 groups consists of the 3 microtubules aligned and fused together so that they appear as 3 circles in a row. Now, the picture that you see here is how a centriole would look like under the microscope. So this is what we call a micrograph. And the descriptions that I mentioned earlier is this. Next, we have the microtubules. Now, the microtubules, as you can see here in the micrograph, are long, slender protein tubes. Those lines as you see here, those are the microtubules. Now, the microtubules form the cytoskeleton of the cell together with the microfilaments. And it is composed of linear polymers, which we call as tubulins. So these linear polymers that you see here, these are what we call as the tubulins, and they make up the microtubules. And they also form the course of cilia and flagella of sperm cells. Microtubules are also important because they play a role in maintaining the cell shape. Next, we have the microfilaments. Now, the microfilaments are important because they help maintain the cell structure as well as its shape. And it's also important in cytoplasmic streaming or cyclosis. And I'll be discussing this further in our synchronous session. Next, we have glycogen granules. So as you can see here, these are glycogen granules. Now, they are abundant in liver cells and they play an important role in the cycle of glucose. Next, we have pigments. Now, pigments are colored parts of the cell which does not require staining because they can be easily seen. They are very much abundant in plant cells. That's why you see that plants have different colors because they have different pigments as well. Now we move on to cell diversity. But before that, I have a question for you guys. Now, how do you think, or how many cells do you think there are in a termite? There are probably more cells than you think. The termite is a relatively complex organism with many specialized cells, but it could not feast on wood without the help of a unicellular protist that live in its digestive tract. Each stowaway protist, in turn, is home to bacteria whose ability to digest cellulose enables both termite and protist to survive. So that's very amazing. Okay, so we have just finished discussing about the organelles and other parts that are common to many cells, but not all cells, take note, are identical. Among the eukaryotes, there are great differences between cells of plants and animals, as you can see here in the picture shown. Now, the picture shows you both plant and animal cells. Now, the red ones are organelles that can be found in animals only, and the green ones are the organelles that you can only see in plants. So these ones, chloroplast and cell wall, can only be seen in plants, whereas centrioles can only be seen in animals. Okay, so one of the most significant differences is that plants have a cell wall. Alright, so another thing is that plants have chloroplasts and plants have vacuoles, or let's just say, to be specific, they have a large central vacuole. Now, the cell wall is a tough, rigid outer covering that protects a plant cell and helps it maintain its shape. The cells of fungi, algae, and bacteria, they also have cell walls. But now, animal cells lack cell walls, but they have other structures such as cytoskeleton for support. So this is how the cell wall of a plant looks like. So there. Next we have the chloroplasts. Now another difference is that plant cells contain organelles called chloroplasts. So these are structures that enable the plant to make sugars through photosynthesis. Now animal cells do not have chloroplasts and cannot make their own food because we know now, or we know of course, that animals get their nourishment by eating other organisms or their products. And this is how the chloroplast would look like. So the entirety of this actually is the chloroplast. So it is made up of the plastoglobule, the thylakoids, the stroma, and of course, it is being enveloped. Next, we have the large central vacuole. So this is a membrane-bound compartment that serves many functions. Now, vacuoles may absorb water, or they may store protein, ions, and the waste products of metabolism. Animal cells have or do not have a large central vacuole, but they may contain smaller vacuoles. So that's why its name is large central vacuole. As you can see here in the picture, it almost takes up the whole cell, like the space of the whole cell. So this from here until here actually is the vacuole. So that's how big the large central vacuole is. Now the table that you see here is a table showing you the comparison between the prokaryotic cells and the eukaryotic cells, particularly animal and plant cells. Which of the organelles are present in eukaryotic cells and prokaryotic cells? Like for instance, we can say that all prokaryotic cells and all eukaryotic cells, they all have cell membranes, whereas in terms of cell wall, although prokaryotic cells and eukaryotic cells both have cell walls, it's only plants and not animals that have cell walls. In terms of nucleus, only eukaryotic cells, both animals and plants, have nucleus. Prokaryotes do not have a nucleus. In terms of chromosomes, prokaryotes only have one circular strand of DNA, whereas animals and plants have many chromosomes. All of these organisms, or all these cells, have ribosomes, although prokaryotic ribosome is different because it only has a small ribosome as compared to eukaryotic cells. Prokaryotic cells also do not have endoplasmic reticulum, Golgi apparatus, lysosome. They also do not have vacuoles and mitochondria, as well as cytoskeleton, as compared to the presence of the said organelles in eukaryotic cells. So that is the summary of it. Now we also have what we call a cell modification and adaptations, and one of which would be the development of microvilli in some cells. Now microvilli, as you can see here in the picture, are actually extensions of the cell membrane. So these are the microvilli. So the function of the microvilli is that because these are extension, it actually increases the surface area of the cell, and in turn the increased surface area enables the cell to have an increased absorption rate of the nutrient. So more microvilli, more surface area, more nutrient absorption. Next, we have the flagellum. As you can see here in the picture, this part here, the tail-like structure, is what we call as the flagellum, and it is intended for locomotion or for the movement of cells. Next, we have root hairs. So the small hair-like structures that you see here, these are root hairs. Now, these are elongated outgrowths from the outer layer of the cells in root, and the same as with microvilli, it increases the surface area of the roots, and because of the increased surface area, there is an increased absorption of water and minerals. And we also have what we call the cell-to-cell junctions, as you can see here. It's very self-explanatory. The name is very self-explanatory, cell-to-cell junction, because it is the point where two cells come together or come into contact with each other. Now, the cell-to-cell junctions are very important because it enables or develops long-term associations, which helps in the formation of tissues and organs.